The architecture of Malus domestica. To comprehend the biological complexity of Malus domestica, one must look past the orchard's aesthetic to the intricate systems that sustain the species. This specimen, named Pomona by the researchers, begins her life cycle beneath the soil surface. Her vitality is anchored by a sophisticated root system. Pomona relies on a primary framework of coarse anchor roots for structural stability, supplemented by a dense, sprawling network of fine, fibrous feeder roots that occupy the upper layers of the soil to maximize nutrient uptake and water absorption. Within Pomona's rhizosphere, a complex symbiosis unfolds. White, thread-like mycorrhizae, specialized fungi, intertwine with her fine roots, extending her reach into the soil to facilitate phosphorus absorption. This subterranean region also reveals the graft union, the point where Pomona's scion meets her rootstock. This distinct lower portion dictates her eventual size, soil adaptability, and resistance to subterranean pathogens, serving as the biological foundation for her entire aerial structure. Rising from the soil, Pomona's trunk serves as a sophisticated transport hub. A cross-section of her mature wood reveals a history of seasonal growth. The outermost layer is the protective bark, an essential barrier against environmental stress. Just beneath this protective shield lies the phloem. This living tissue is responsible for the downward translocation of photosynthates, sugars produced in the leaves, ensuring that energy reaches her roots and lower tissues. The interface between Pomona's phloem and her interior wood contains the vascular cambium, a microscopic layer of meristematic cells. This is the engine of her secondary growth, constantly dividing to produce new phloem toward the exterior and new xylem toward the interior. The xylem, or sapwood, constitutes the bulk of her trunk. It serves as the primary conduit for water and dissolved minerals, pulling them upward from the roots to the highest leaves through a process driven by transpiration. As Pomona's branches extend into the canopy, her growth patterns are dictated by bud morphology. She bears vegetative buds, which produce only leaves and shoots, and mixed buds, which contain both embryonic leaves and flowers. As a spur-bearing cultivar, Pomona produces much of her fruit on short, aged wood known as fruiting spurs. These condensed shoots represent years of potential energy, concentrated into plump, fuzzy terminal buds. Pomona's foliage represents her primary site of energy production. Each vibrant green leaf is an architectural marvel of gas exchange and light capture. On the leaf's underside, thousands of microscopic stomata act as regulated gateways, allowing carbon dioxide to enter for photosynthesis while releasing oxygen and water vapor. Inside her mesophyll cells, chloroplasts containing chlorophyll capture solar energy to synthesize the glucose that fuels her growth. By mid-spring, Pomona produces a cyme-like inflorescence. The central flower in each cluster is known as the king bloom. She ensures this bloom opens first. If successfully pollinated, it has the potential to produce the largest fruit in the cluster. The surrounding lateral flowers follow in a predictable sequence, creating a tiered display designed to maximize the window for cross-pollination by visiting insects. The anatomy of Pomona's individual flowers is a study in reproductive precision. Each blossom features 5 sepals at the base and 5 petals. At the center, she presents numerous stamens, each topped with a pollen-bearing anther. These surround the central pistil, which consists of 5 styles leading down to an inferior ovary. It is within this protected ovary that the future seeds reside, waiting for the genetic contribution of pollen to begin their development. As the season progresses, the ovary transforms into a pome. The fruit of Malus domestica is actually the expanded hypanthium or floral tube, which has swollen to enclose the true fruit. When sliced vertically, Pomona's internal anatomy is revealed. The central core is the botanical ovary, consisting of 5 cartilaginous carpels that house the seeds. The distinct core line is the remnant of the vascular bundles that once nourished the developing ovary. The existence of Malus domestica is a testament to botanical synergy. From the mycorrhizal associations in the dark soil to the stomata in the sunlit leaves, every structure of Pomona works in concert to ensure her survival and reproduction. She is not merely a source of harvest, but a complex biological system, a masterwork of evolutionary engineering that continues to thrive through the delicate balance of vegetative health and reproductive success.